The thermodynamic advantage of metabolic exploitation. Whether manifested through social parasitism, brute theft, or structural labor coercion, exploitation provides a highly efficient evolutionary shortcut in the biospheric ledger. This parasitic lifestyle allows an exploiting lineage to completely eliminate the vast metabolic overhead costs of survival by systematically offloading them onto a vulnerable host population. Every independent species is bound to a high maintenance budget requiring immense daily outputs of cellular energy, ATP, to forage for nutrients, construct physical defenses, and regulate internal cellular health. This high baseline expenditure naturally places a strict ceiling on the energy a lineage can allocate toward a direct reproductive output. By transitioning to an exploitative strategy, social parasites like the specialized slave-making ant Polydurus systematically shed these capital overhead costs. Over deep evolutionary time, these insects have completely lost the morphological structures and behavioral pathways required to excavate nesting chambers, seek out food sources, or feed their own larvae. By outsourcing with a near-zero maintenance budget, this systematic freedom allows the parasite to channel its entire metabolic energy reserve into producing high-quality, behaviorally dominant reproductive elites, vastly outbreeding and out-expanding surrounding independent species. Furthermore, this thermodynamic theft enables a process known as evolutionary niche hijacking. Cultivating, adapting to, and securing a novel ecological niche requires millions of years of gradual genetic modification and intense selective pressure. Exploitative species bypass this immense timeline entirely by targeting and hijacking a mature, highly stable niche that has already been perfected by another organism. This systemic infiltration relies on locating and hacking a sensory loophole within the host's evolutionary architecture. Exploitative lineages survive by mapping and systematically triggering the predictable, hard-coded behavioral routines of their targets, utilizing specialized chemical signals or vocal mimics that the host is genetically programmed to nurture. Because the host population cannot rapidly rewrite its core genetic instincts without incurring a catastrophic evolutionary lag, the exploiter secures a long-term, high-utility resource stream at a minimal metabolic cost. This dynamic provides a vivid biological parallel to the resource-heavy Homo colossus anomaly, where technological systems hijack the ancient, self-regulating loops of the Gaia organism to extract energy without contributing to planetary stability. Organized conflict and systemic exploitation are not uniquely human traits, but are highly conserved, evolved strategies that appear across diverse kingdoms of life. For billions of years, intense resource scarcity and the mathematical realities of genetic relatedness have driven non-human organisms to develop complex group violence and structural parasitism. Far from being moral anomalies or cultural inventions, these behaviors represent optimal thermodynamic paths to maximize reproductive fitness. By treating armies, slave-making castes, and behavioral manipulation as metabolic shortcuts rather than conscious choices, we can dismantle the anthropomorphic trap and analyze the universal mechanics of biological exploitation on a purely objective ledger. The mechanics of formicid strategy. Formicid warfare represents the peak of non-human organized combat across the planetary biosphere. Because ant colonies operate as cohesive superorganisms governed by an asymmetrical haploid relatedness coefficient, they successfully bypass the individual survival instinct that limits mass coordination in solitary species. This intense genetic alignment allows them to execute highly structured, group-level military strategies that directly mirror advanced human tactical maneuvers. Rather than relying on a conscious command architecture, these complex military operations are driven entirely by dynamic chemical communication networks and hard-coded evolutionary algorithms, converting thousands of individual units into a single, flowing kinetic force. This formicid strategy unfolds not as a chaotic brawl, but as a highly regulated kinetic conflict continuously managed through real-time chemical feedback loops. The operational phase is initiated when specialized scouts locate an enemy border or an encroaching foraging column, prompting the immediate release of volatile trail pheromones along the substrate back to the home nest. This chemical recruitment operates via precise gradient dynamics. The intensity and volatility of the deposited pheromone trail directly correspond to the scale of the encountered threat. This concentration gradient regulates the recruitment velocity, dictating the exact volume and rate of reinforcement troops deployed to the front line. Once deployed, the colony utilizes extreme morphological role specialization through its hard-coded caste system. The minor workers function as the frontline infantry, being metabolically cheap to produce and highly agile. They deploy in dense, high-density skirmish lines to overwhelm the enemy through sheer numerical superiority. Their primary tactical objective is to physically pin down larger adversaries, immobilizing their limbs while simultaneously spraying their reticular membranes with toxic, highly concentrated formic acid. This coordinated restraint allows the major workers and specialized soldiers, the heavy armor of the superorganism, to advance safely. Equipped with massive, highly muscled mandibles, these major soldiers act as heavy breachers and choke point blockers, applying crushing physical force to sever enemy armor and terminate targets that have been successfully immobilized by the minor infantry. Tactical battle formations and group dynamics. The genus Phytol, colloquially known as big-headed ants, alongside various highly territorial Formica species, demonstrate advanced tactical flexibility, altering their battle formations dynamically to adapt to shifting battlefield kinetics. During territorial disputes along established colony borders, Phytol forces execute careful line assessment to evaluate the relative demographic strength of the opposition. If the defenders detect that the invading army possesses superior numbers, they immediately abandon open ground skirmishes and strategically restrict the engagement to narrow subterranean tunnels, root matrices, or tight rock crevices. This maneuver implements classic phalanx mechanics by packing major soldiers shoulder to shoulder inside a confined tunnel mouth. The defending colony effectively neutralizes the enemy's numerical advantage. This spatial restriction forces the conflict into a series of manageable, isolated one-to-one battles, allowing the superior defensive armor of the major caste to hold the perimeter against overwhelming odds. When a defensive line is ultimately overwhelmed by sheer force or chemical subversion, the superorganism does not collapse into uncoordinated panic. Instead, it executes a strategic retreat driven by chemical alarm shifting. As frontline workers are crushed or mortally wounded, their venom glands and mandibular reservoirs release intense concentrations of highly volatile panic pheromones, such as undecane or specific alert ketones. When this chemical cloud crosses a critical threshold, neighboring workers respond to the localized signal by pulling back from the immediate conflict zone. This strategic withdrawal is followed by structural engineering maneuvers. The retreating workers rapidly seal the inner tunnel, followed by structural engineering maneuvers. The retreating workers rapidly seal the inner tunnel entrances with soil and detritus, creating a secondary defensive perimeter closer to the maternal nest. This tactical resilience is frequently paired with the aggressive physiological warfare, vividly illustrated by the fire ant, Solenopsis invicta. During mass engagements, these ants deploy solenopsin, a highly toxic piperidine alkaloid-based venom, administered via abdominal stingers. This chemical weapon induces rapid neuromuscular paralysis in rival arthropods, systematically destroying the enemy's coordinated defensive formations by knocking out individual workers and breaking their chemical communication loops. The strategic objective: demographic and resource extermination. Ultimately, formicid warfare is driven by a strict ecological imperative to secure vital foraging territory and systematically eliminate adjacent genetic competitors. As a superorganism expands its population density, its operational foraging radius inevitably collides with the borders of neighboring colonies. Transforming spatial proximity into an intense survival pressure, this territorial encroachment escalates from minor resource competition into total war, which functions as a spatial calculation to secure high-energy food patches, aphid livestock colonies, and long-term nesting space. Once a front line is successfully broken and the secondary defensive perimeters are breached, the attacking columns transition into an invasive formation pushing deep into the core chambers of the defeated nest. The primary strategic objective of this infiltration is a definitive decapitation strike, the localization and termination of the rival queen. Because the queen serves as the sole reproductive engine of the superorganism, her destruction immediately halts the generation of new workers, inducing an irreversible demographic collapse. Following the termination of the queen, the victorious colony pillages the nest, seizing the remaining larval and pupal brood to extract their nutritional value or integrate them into their own labor force, permanently erasing the rival genetic lineage from the ecosystem. This unyielding, resource-driven extraction provides the clear biological precursor to the runaway exploitation seen in Homo colossus, where industrial and military systems similarly dismantle natural perimeters to harvest the resources of the planet. Infochemical sabotage and psychological hijacking. If physical morphology represents the kinetic weaponry of formicid conflict, chemical synthesis serves as a strategic missile and cyber warfare capability. Because ants do not navigate combat through visual feedback, they operate within a complex, fluctuating olfactory landscape where structural communication is dedicated by molecular signals. By isolating, synthesizing, and deploying volatile organic compounds, warring colonies can manipulate enemy behavior, disrupt tactical communication lines, and deliver lethal physiological payloads directly to the front line. The most sophisticated manifestation of this chemical warfare is infochemical sabotage, which utilizes propaganda allomones to actively rewrite the behavioral software of opposing forces. The physiological hijacking is vividly demonstrated during the raiding vectors of the slave-making genus Polyergus. During an assault on a host formica nest, the raiders release massive quantities of volatile aldehydes and ketones from their highly hypertrophied Dufour's gland. This concentrated chemical cloud floods the antennal olfactory receptors of the defending host population, inducing complete sensory saturation and effectively blinding their communication network. Bypassed by this infochemical disruption, the host workers are unable to coordinate defensive formations. Instead, they enter a state of frantic, uncoordinated disorientation, allowing the Polyergus raiders to breach the inner nursery chambers with minimal physical resistance. This tactic is augmented by alarm signal mimicry, a form of sensory sabotage deployed by rogue species such as the thief ant, Solenopsis fugax. While raiding adjacent nests to pillage larval resources, these specialized social parasites spray unique alarm pheromone mimics directly into the host tunnels. The host workers mistake these synthetic allomones for their own internal distress signals, triggering an immediate localized panic cascade. This sensory deception causes the defenders to prematurely evacuate their chambers, or misidentify their own nestmates as hostile invaders, turning the colony's collective defense apparatus against itself. Corrosive hydrocarbons and physiological delivery. Beyond behavioral manipulation, formicid systems deploy specialized chemical payloads designed for direct tissue destruction. neuromuscular paralysis, and localized mortality. Within the subfamily Formicinae, this chemical artillery is anchored by the mass synthesis of formic acid, a simple carboxylic acid stored in highly concentrated abdominal reservoirs. During frontline skirmishes, workers execute a distinct kinetic maneuver, pivoting their gasters forward between their pelvic limbs to spray a precision stream of atomized acid directly into the opposing range. At the molecular level, this carboxylic acid acts as a powerful solvent that rapidly corrodes the protective epicuticular wax layer of the target insect's exocuticle. The destruction of the hydrophobic barrier induces immediate desiccation and severe localized tissue necrosis. This corrosive artillery is complemented by the deployment of alkaloid neurotoxins, most notably the Solenopsis synthesized by fire ants, Solenopsis species. Solenopsins are specialized piperidine alkaloids characterized by a highly hydrophobic molecular structure, an evolutionary adaptation that allows the toxin to rapidly penetrate the lipid and chitin layers of an opponent's outer cell. Once inside the hemolymph, solenopsins act as a potent necrotic and neurotoxic agents, directly disrupting cell membrane permeability and inhibiting mitochondrial cellular respiration. This double-pronged attack triggers rapid systemic paralysis and localized cell death in opposing forces within minutes of injection. To maximize the efficiency of these physiological payloads, certain wood ant species utilize an integrated acid-binding fixer strategy. Workers use their mandibles to inflict deep physical punctures into the articular membranes and joints of an enemy's limbs, and then immediately spray their formic acid payload directly into the open wound. This behavioral combination completely bypasses the protective outer exocuticle, delivering the toxic compound straight into the open hemolymph to induce rapid systemic shock and immediate metabolic collapse. Cuticular hydrocarbon CHC, spoofing and stealth operations. The foundational matrix of all social order within a eusocial colony is its cuticular hydrocarbon CHC profile, a highly specific inherited blend of linear alkylanes, branched alkylanes, and alkenes coating the outer exocuticle that functions as a chemical passport. Ant colonies utilize their antennal chemoreceptors to scan this lipid layer, executing rapid nestmate recognition to isolate and eliminate non-kin intruders. To bypass this security system, parasitic queens and specialized raiders have evolved CHC spoofing, a chemical stealth operation that allows them to actively synthesize or behaviorally acquire the exact hydrocarbon signature of their target host species. This chemical camouflage facilitates a highly effective Trojan horse strategy during the colony infiltration phase. By matching the host's specific cuticular hydrocarbon blend, an invading parasitic queen can walk directly through the defensive perimeters of a hostile nest entirely undetected. The sensory receptors of the defending workers identify her as a biological sister, allowing her to navigate the subterranean tunnels unmolested until she locates and executes the resident host queen. Following this decapitation strike, the parasite queen absorbs the maternal chemical cues of the dead monarch, establishing a multi-generational cloak. The host workforce remains completely unaware of the genetic replacement, continuously expending its collective metabolic energy and cellular ATP to rear the offspring of a genetic competitor. This systemic hijacking of a mature host infrastructure provides the clear biophysical parallel to the resource-heavy Homo colossus footprint demonstrating how human technological intervention overrides the self-regulating communication loops of the biosphere. The mechanics of the Myrmecocytus tournament matrix. While formicid conflict often results in absolute demographic annihilation, certain specialized genera have evolved non-lethal evaluation strategies to manage geographic resource boundaries. When the projected energetic cost of a total warfare outbalances the potential caloric or territorial gain, natural selection favors the implementation of ritualized tournaments. These interactions function as structured, real-time behavioral audits, allowing neighboring colonies to precisely evaluate an opponent's demographic strength, population density, and resource reserves without incurring physical fatalities or destroying their respective workforces. The premier baseline for this non-lethal strategic assessment is found with honeypot ants of the genus Myrmecocytus, specifically Myrmecocytus mimicus. The tournament matrix is initiated when foraging scouts from two distinct non-kin nests collide in a high-value resource zone. Rather than engaging in immediate lethal combat, these scouts release specialized recruitment pheromones that draw hundreds of worker elites to a neutral centralized tournament field. Upon arrival, the opposing forces face each other and execute a highly specific behavioral display, elevating their physical profiles by fully straightening their legs, a maneuver known as stilt walking, while simultaneously raising their gasters vertically. By artificially amplifying their physical dimensions, individual workers display their precise structural scale. Because larger physical size correlates with an evolutionary advantage, this stilt walking display allows opponents to gauge the physical maturity of the competing colony's workforce. This aggressive communication is augmented by ritualized body buffeting and parallel walking maneuvers, where ants drum their antennae against one another to evaluate the chemical profiles and physical resistance of their opponents without inheriting lethal jaw snapping or chemical spraying. The strategic calculus: parity versus asymmetry. The ultimate evolutionary purpose of a ritualized tournament is to calculate the mathematical probability of a military victory before investing finite metabolic capital in the actual warfare. This behavioral matrix operates as a biological computer, processing real-time sensory inputs to determine whether a conflict will yield a net fitness benefit or a catastrophic resource deficit. When two highly stable, mature colonies interact, they frequently achieve a condition of parity. If both sides continuously deploy an equal density of high-quality stilt-walking workers to the tournament grounds, the interaction can persist for several days without escalating into physical violence. The persistent sensory feedback informs the internal algorithms of both superorganisms that a hot war would result in mutual destruction, presenting a high fitness cost for a low resource return. Consequently, the tournament naturally dissolves and both colonies execute a mutual tactical retreat to establish a stable, shared territorial boundary equidistant between their respective nests. Conversely, if one colony is experiencing a demographic decline or a severe internal resource shortage, it will fail to supply a sufficient volume of high-quality workers to the tournament field. The opposing workers instantly detect this structural deficit, mapping the drop in the opponent density and the smaller physical stature of the remaining forces. The moment this asymmetry is verified, the ritualized tournament structure collapses. Dropping their non-lethal posture, the dominant colony transitions into an immediate, coordinated invasion. They march directly back to the vulnerable nest of the weaker colony, bypass its broken defenses, execute the resident queen, and plunder the internal brood and fluid-filled honeypot repletes. This flash transition demonstrates the ritualized behaviors are not driven by peaceful intent, but by a ruthless calculation of strategic advantage. Evolutionary advantages of non-lethal auditing. Ritualized tournaments represent a highly optimized evolutionary solution to the problem of territorial defense, under the constraints of the economic defensibility model. Within a superorganism, worker and soldier elites are highly valuable metabolic assets. Their production requires a substantial investment of collective energy during the prolonged egg, larval, and pupal stages. Wiping out thousands of these specialized units in an unmitigated hot war over a minor foraging patch reduces the colony's overall carrying capacity and leaves it vulnerable to secondary predators. Tournaments allow the superorganism to protect this substantial labor investment, settling boundary disputes via a behavioral proxy that preserves its active workforce for essential foraging and nursing duties. Furthermore, these interactions function as an effective, open-source intelligence mechanism. By engaging in a low-risk physical display, a superorganism can continuously update its internal mathematical mapping of the local competitive landscape without risking a systemic security failure. This continuous feedback loop allows the colony to dynamically adjust its foraging boundaries based on real-time strength of its neighbors, optimizing its resource extraction while maintaining a safe defense profile. The sophisticated balance between resource assessment and calculated survival provides a stark contrast to the runaway, uncoupled destruction that characterizes Homo colossus. While natural superorganisms utilize non-lethal auditing to remain within biospheric limits, modern human systems routinely override these regulatory loops, generating the deep-seated transgenerational trauma outlined in my unified hypothesis. The Raider morphology, engineered for combat, dependent on slaves. Doulosis, or obligatory slave-making behavior, represents a highly specialized behaviorally aggressive form of social parasitism found within specific vermicid lineages. Within this evolutionary framework, the parasitic species has completely shed its metabolic maintenance overhead, systematically outsourcing the tasks of nest construction, territory foraging, resource defense, and brood nursing onto an enslaved workforce. To sustain this exploitive architecture, specialized raider castes combine advanced military group maneuvers with the sophisticated biochemical deception to launch regular, targeted assaults on adjacent host colonies, harvesting their mature brood to continuously refresh their internal captive labor supply. This parasitic lifestyle is driven by extreme morphological adaptations designed exclusively for high-force combat, creating a profound biological trade-off that locks the species into an absolute state of host dependency. This morphological specialization is vividly illustrated by the razor-sharp, scythe-shaped mandibles of the Amazon ant, Polyergus, raider caste. Unlike standard worker ants, which possess multi-purpose serrated and spatulate jaws optimized for soil excavation, seed processing, and the delicate handling of microlarvae, the mouthparts of Polyergus workers have been reduced to smooth, sickle-shaped biological daggers. These specialized weapons are biomechanically optimized for high-force piercing, allowing the raiders to puncture the heavily sclerotized head caps and thoracic plates of opposing ants with surgical precision. However, this extreme combat optimization introduces a severe metabolic trap. These smooth, curved mandibles lack the structural surfaces required to manipulate soil particles, grasp microscopic eggs, or collect independent nutrients. Consequently, Polyergus raiders are anatomically and behaviorally incapable of maintaining their own nests or nursing their own offspring. Furthermore, they are unable to feed themselves independently without an enslaved host workforce to actively regurgitate liquefied carbohydrates and proteins directly into their mouths via the process of trophallaxis. A Polyergus colony would rapidly starve to death within its own fortress, surrounded by its own specialized weaponry. This is the strategic sequence of the Dulotic raid. A dulotic raid functions as a precisely timed, high-velocity military operation that coordinates scouting intelligence, rapid mass recruitment, and intense chemical propaganda to systematically overwhelm host defenses while minimizing raider casualties. This offensive sequence is initiated by a rigorous reconnaissance phase, during which single specialized scouts locate mature nests of target host species, such as Formica subcerriaceae. The scout evaluates the precise geographic coordinates and developmental maturity of the host community, and then executes a high-speed recruitment run back to the parasitic home nests. Upon arrival, the scout releases a volatile, highly attractive recruitment pheromone from her hindgut reservoir. Within minutes, this intense chemical signal prompts hundreds of raider elites to stream out of the nest entrance, organizing into a dense, fast-moving column that marches directly along the established pheromone trail toward the target host community. When the advancing polyergus column reaches the outer perimeter of the target Formica nests, they do not rely on physical force alone. Instead, they execute a devastating form of chemical warfare, deploying propaganda allomones from their highly hypertrophied Dufour's gland. This concentrated chemical cloud releases a mix of volatile aldehydes and ketones that completely floods and oversaturates the host ant's antennal olfactory receptors, including immediate sensory sabotage. This infochemical disruption triggers a localized panic cascade within the host colony, blinding their internal alarm systems and causing host workers to break their defensive phalanxes and scatter into the surrounding vegetation. This tactical disorientation allows the raiders to execute a rapid kinetic infiltration into the subterranean tunnels with minimal resistance. Any host defenders that manage to overcome the chemical confusion and attempt to guard the perimeters are quickly eliminated by the raiders, who use their sickle-shaped jaws to pierce the central nervous systems of the defenders. Once inside, the raiders ignore the host food storage chambers and focus entirely on logistics, infiltrating the deep nursery chambers to selectively harvest mature host pupae and older larvae. The raiders grasp the captured pupae gently within their curved jaws to prevent mechanical damage, and then immediately reverse their direction, retreating back along the pheromone trail to transport the stolen biological capital back to their own fortress. The integration phase. The psychological hijack. The ultimate long-term success of a dulotic system relies on exploiting a hard-coded, highly predictable vulnerability in insect neurological development: post-eclosion chemical imprinting. This integration phase executes a profound psychological hijack, converting the stolen host generation into a compliant, self-regulating workforce without the application of physical restraint or visible coercion. When a standard worker ant undergoes eclosion, hatching from its protective pupal case, its newly formed outer cuticle lacks a fixed, stable blend of lipids and waxes, known as the cuticular hydrocarbon profile. Over the initial 48 to 72 hours of its adult life, the newly emerged worker records and seals the specific olfactory signatures of its immediate microenvironment, permanently hard-coding these environmental odors as the baseline definition of home and kin. Because these stolen Formica pupae complete their eclosion inside the raider nest, they imprint on the raider's unique chemical signature, mistaking the parasitic colony for their own genetic family. This false kinship completely realigns the host worker's behavioral software. Upon reaching the physical maturity, these enslaved workers naturally take over the daily operation of the parasitic nest. They excavate new tunnels, forage for environmental nutrients, defend the outer perimeters against non-kin intruders, and feed and nurse the raiders' offspring, completely unaware that they are investing their entire lifetime budget of cellular ATP into a genetic dead end that wiped out their true biological family. The systemic hijacking of a mature host infrastructure provides a vivid biological parallel to the resource-heavy Homo colossus footprint outlined in my unified hypothesis. Just as the polyergus parasite exploits the hard-coded behavioral loops of the host to harvest its energy, modern industrial systems override the self-regulating feedback loops of the Gaia organism to extract planetary resources. This process drives the living system away from its native cooperative equilibrium and anchors the deep-seated transgenerational trauma that my report aims to diagnose and resolve. The hostile entry, overcoming the barrier of aggression. The foundation of a long-term parasitic or zootic colony relies on an initial high-risk act of environmental espionage, colony usurpation. A parasitic queen cannot build a workforce independently, lacking the morphological adaptations and behavioral software required to excavate a nesting architecture or nurse her initial brood. Instead, she must successfully infiltrate an established hostile host nest and seize control of its existing labor force. This infiltration requires overcoming a defensive perimeter maintained by specialized guard workers who continuously sample the particular hydrocarbon profile of any insect attempting to enter the nest. Under baseline conditions, these guards immediately tear apart individuals with unfamiliar chemical signatures, mandating that the invading queen utilize precise chemical camouflage to shift her molecular profile from an obvious intruder to an accepted ruler. This chemical subversion progresses through two primary evolutionary modalities: kinetic usurpation and passive chemical disguise. In the hyper-aggressive usurper strategy practiced by newly mated polyergus queens, the queen begins her infiltration by executing a solitary ambush on a lone host worker near the nest perimeter. Utilizing her scythe-shaped mandibles, she executes the worker with a targeted puncture to the central nervous system, and then systematically licks, grooms, and rolls against the fresh carcass. This precise tactile behavior facilitates the direct mechanical transfer of the dead worker's surface lipids and waxes onto her own outer exocuticle, creating an immediate, temporary chemical disguise that permits her to walk past the other guards undetected. Conversely, passive stealth infiltrators, such as parasitic queens of the genus Lagius, rely on chemical sedation rather than immediate physical execution. As the Lagius queen approaches the host nest entrance, she releases volatile neural calm secretions from her mandibular glands. These airborne compounds act as localized sedatives or confusion alamodes, actively suppressing the olfactory receptors and aggressive behavioral responses of the host guards. This temporary drop in aggression generates a localized scrutiny blind spot, allowing the queen to slip past the outer defenses completely unnoticed without triggering the colony's defensive alarm cascade. The regicide phase, executing the host monarch. Once the evading queen successfully navigates the outer defensive perimeter and enters the subterranean nest tunnels, she transitions into the regicide phase, tracking down and eliminating the resident host monarch. If the host queen remains alive, her persistent pheromonal output will continue to produce loyal host workers who will eventually detect the structural anomalies of the invader and destroy her in a wave of delayed aggression. To prevent this scrutiny failure, the invader utilizes internal chemical trails and structural heat gradients to navigate the dark, complex tunnel networks, locating the host queen's inner royal chamber with high spatial precision. Upon entering the royal chamber, the invader launches a swift, surgical assault, leveraging her specialized jaw architecture to pierce the head caps of cervical membranes of the host queen. After the execution, the invader stays with the dead queen for several hours or days, continuously grooming the carcass and bathing in its leaking exuvial and internal fluids. This post-lethal behavior allows the invader to absorb the host queen's highly refined blend of royal pheromones, permanently updating her chemical profile. By incorporating these really optimized cuticular hydrocarbons into her outer shell, the invader subverts the host workforce's recognition matrix, ensuring that the surrounding attendants mistake her for their true maternal leader. Long-term subversion, managing the host workforce. Following the successful execution of the host queen, the invader must transition from temporal mechanical camouflage to long-term biosynthetic subversion. This ensures that the remaining host workers accept her as their true biological mother, rather than destroying her as the initial chemical disguise begins to decay. Over the days following the regicide, the parasitic queen's internal metabolism shifts, She ceases to rely on stolen surface lipids and begins actively synthesizing the exact complex CHC profile of the host colony within her postpharyngeal and Dufour's glands. The host workers read the synthetic profile through antennal contact, identifying her as their true genetic mother and fully integrating her into the social architecture of the nest. To maintain this absolute control, the parasitic queen releases powerful chemical signals that suppress the reproductive capacity of her hosts. These royal primer pheromones actively downregulate the ovarian development of the remaining host workers, ensuring they remain functionally sterile. This chemical domination forces the host workers to spend their remaining adult life cycles focusing their entire metabolic energy and cellular ATP budget on caring for the invader's eggs, turning the host colony into a self-contained production factory for a genetic competitor. This multi-layered systemic hijacking provides the final biological anchor. It illustrates how natural organisms use chemical and behavioral engineering to manipulate existing infrastructures for survival. This sets up a powerful contrast with the emergence of the resource-heavy Homo colossus. I will return to this in Phase 2, where we will explore how human consciousness broke away from these natural balances due to the ancient transgenerational trauma. Breaking the chemical illusion, detecting the parasite scent. Enslaved hosts do not always remain passive tools of their captors. Over deep evolutionary time, host species have developed a quiet, highly destructive evolutionary counter-strategy known as slave rebellion. This behavior represents a subtle form of evolutionary sabotage where enslaved workers, despite being chemically deceived into maintaining the parasite nest, routinely identify and execute the developing offspring of their captors. This targeted culling raises the metabolic cost of raiding and puts a strict biological brake on the expansion of slave-making colonies. The success of this strategy depends entirely on the host workers' ability to overcome their chemical conditioning and differentiate between their own species and the parasite lineage. During the larval and pupal stages, parasite offspring must develop their own species-specific reticular hydrocarbon profiles to prepare for adulthood. Enslaved Formica workers constantly inspect the nursery using their antennae, sampling these lipids. While adult parasite queens can successfully mimic host smells, the developing pupae often produce distinct, unspoofed chemical signatures. This chemical mismatch gives the enslaved workforce a critical window of opportunity, allowing them to spot the genetic intruders resting inside their own nursery chambers. Mechanics of sabotage. The invisible labor strike. Once the enslaved workers identify the parasite pupae, they do not launch an open violent revolt against the heavily armed adult raiders. Instead, they engage in quiet, highly effective systemic sabotage, splitting their tactics between passive neglect and active infanticide. Through passive neglect, enslaved workers simply stop feeding and grooming the parasite larvae. They actively move the parasite pupae away from warm, humid incubation zones and leave them in cold, damp, or moldy corners of the nest, letting natural exposure and starvation destroy them. When they pivot to active infanticide, workers tear open the protective silk cocoons of the parasite pupae before they can fully mature. Once the pupa is exposed and defenseless, the enslaved workers bite off its limbs or chew through its soft skin, subsequently consuming the dead pupae or throwing the remains out of the nest as waste to destroy the parasite's next generation. The evolutionary impact, limiting parasitic success. The slave rebellion strategy acts as a powerfully evolutionary check, keeping slave-making populations in balance and preventing them from completely wiping out local host communities. Field studies show that rebellious host workers can systematically destroy up to 2/3 of a slave-making colony's female pupae, severely limiting the number of new raiders available for future missions. By destroying the parasite's offspring, the enslaved workforce loses the return on investment of the original raid. The adult parasites waste massive amounts of energy capturing host pupae, only to have those hosts turn around and wipe out the parasite's genetic future. While a rebellious worker cannot save its own life or pass on its individual genes, its sabotage directly protects neighboring, non-enslaved host colonies. By weakening the parasite fortress from within, The slaves lower the overall raid pressure on the local environment, ensuring the long-term survival of their wild genetic relatives. This evolutionary resistance demonstrates how even deeply hard-coded exploitation loops face systemic checks, mirroring my unified hypothesis. By showing that true eusocial balance actively works to suppress runaway parasitic extraction. The biochemical ledger of allelopathic weaponization. Allelopathy represents the principal mechanism of organized, high-density resource and chemical warfare within the kingdom Plantae. Because plants are sessile organisms incapable of physical locomotion, natural selection has engineered their offensive and defensive strategies entirely within a biochemical and thermodynamic framework. Far from being passive components of the landscape, plant communities engage in continuous, active territorial expansion and demographic displacement through the synthesis and deployment of toxic, secondary metabolites designed to inhibit, stun, or execute neighboring rival species. This strategy operates as an aggressive expansionist program, utilizing specialized chemical payloads to clear microhabitats, secure absolute access to groundwater and soil nutrients, and monopolize solar radiation. Plants destroy these sophisticated chemical weapons through four primary ecological vectors designed to infiltrate the surrounding environment. Through rhizosphere exudation, root tissues actively pump toxic compounds directly into the adjacent soil matrix, altering the biochemical composition of the immediate underground environment. This is reinforced by leaching, a process where rain, dew, or fog washes water-soluble phytotoxins off leaf surfaces, creating highly concentrated toxic perimeter beneath the plant's canopy. Simultaneously, plants utilize volatilization, releasing volatile aromatic compounds through their stomatal openings in the surrounding air to form a heavy chemical vapor cloud that eventually settles into the local topsoil. Finally, plants execute a delayed tactical strike through decomposition, intentionally storing toxic metabolites in senescent leaves. When these leaves drop and decay, they permanently poison the topsoil layer against competing seedlings. At a molecular level, these allelopathic chemicals do not merely crowd out competitors. They actively disrupt core cellular machinery to cause metabolic and structural collapse. Through mitotic inhibition, compounds like juglone or specific volatile terpenes arrest cellular division within foreign root tips, preventing target seedlings from establishing a structural foothold in the soil. Concurrently, these compounds introduce photosynthetic disruption by degrading chlorophyll pigment of permanently blocking electron transport chains within the chloroplasts of neighboring plants, starving them of light harvesting capacity. This internal starvation is accelerated by membrane peroxidation, wherein advanced phytotoxins alter the permeability of target root membranes. This molecular breach induces rapid, unmitigated ion leakage and destroys the precise osmotic balance required for water absorption, naturalizing the competitor's survival capacity. Case studies of botanical invasions and territory monopolization. The real-world execution of these botanical warfare strategies is vividly illustrated by specific invasive and dominant flora taxa that establish absolute territorial monopolies. The black walnut tree serves as the classic empirical baseline for targeted, localized biochemical exclusion zones, deploying an environmental filter that protects its resource base. The tree synthesizes a non-toxic chemical precursor called hydrojuglone within its leaves. Roots and bark tissues. When hydrojuglone is excreted into the soil matrix via root tissues or leached from fallen leaves, it undergoes rapid oxidation upon contact with air or soil microbes, transforming into the highly toxic quinoid compound juglone. 5-hydroxy-1,4-naphthoquinone. Juglone forms a permanent biochemical death zone stretching up to 50 to 80 feet from the trunk, targeting key enzymes in competing species such as tomatoes, apples, and birch trees. By inhabiting plasma membrane pumps and halting cellular respiration, juglone introduces severe plasmolysis and mitotic arrest, ensuring that no rival vegetation can survive to steal groundwater or solar radiation. This chemical domination takes an aggressive physical turn in eucalyptus forests, which utilize a dual-threat chemical and physical warfare strategy to permanently alter entire ecosystems through pyrophytic fire warfare. The leaves of the eucalyptus tree are densely packed with highly volatile, flammable terpenoid oils, such as eucalyptol. During periods of high ambient temperature, these oils vaporize through the stomata, creating a dense, highly explosive aerosol cloud across the forest canopy. Eucalyptus does not avoid wildfires, it actively encourages them, utilizing its volatile oil cloud to turn minor lightning strikes into catastrophic, high-intensity canopy fires. This creates a severe evolutionary asymmetry. While the intense heat completely obliterates native, non-pyrophytic trees and the subterranean sea banks, the eucalyptus survives undamaged, protecting by insulated, deep dark epicormic buds and the underground lignotubers. Following the blaze, the charred soil becomes a rich, nutrient-dense seedbed for newly dropped eucalyptus seeds, allowing the species to completely displace the original plant community and monoculture the landscape. The thermodynamic and evolutionary trade-offs. Like formicid armies or dulotic raiding parties, maintaining an allelopathic chemical arsenal requires a significant, continuous, energetic investment within the biospheric ledger. Synthesizing complex, carbon-heavy phenolics, alkaloids, and volatile terpenes demands a substantial diversion of cellular ATP and fixed sugars away from primary somatic growth and direct reproductive output. A plant utilizing an allelopathic strategy must therefore achieve a high enough return on resource acquisition, gaining exclusive access to localized water tables, nitrogen reserves, and light gaps. To offset the continuous manufacturing tax, if the environmental conditions change and resources become abundant, this high metabolic overhead can become a severe fitness deficit, allowing non-allelopathic wild-type strains to outgrow and outcompete the chemical specialists. To survive this intense chemical output, plants must simultaneously develop complex internal defensive mechanisms to avoid autotoxicity, the catastrophic scenario of poisoning themselves with their own weapons. To resolve this structural problem, plants store their active toxins as inert, non-toxic glycoside. Precursors, which are only converted into their active states upon excretion into the environment or contact with extracellular enzymes. Alternatively, plants isolate these active chemicals inside specialized wax-lined glandular trichomes or intracellular vacuoles, keeping the toxic compounds physically segregated from the cell's own sensitive metabolic machinery until the exact moment of deployment. This sophisticated balance between self-preservation and calculated environmental extraction provides the final cross-kingdom anchor for Phase 1 of my report. It shows that even sessile, non-conscious life forms utilize highly calculated, resource-driven strategies to navigate scarcity. preparing you for Phase 2, where we will diagnose how the human-specific anomaly broke away from these natural balances. Rhizosphere intelligence, self versus non-self recognition. If allelopathy represents the long-range artillery of botanical conflict, the rhizosphere serves as the close-quarters trench network where underground territory is won or lost. Beneath the soil surface, plant root systems engage in highly dynamic, aggressive territorial competition. Far from being passive structural anchors, roots function as sophisticated sensory platforms. They actively scan the chemical profiles of the soil matrix, identify the genetic signatures of adjacent roots, and deploy targeted growth strategies to outmaneuver, starve, or physically strangle rival root architectures. The baseline requirement for this subterranean combat is the capacity to accurately classify surrounding biological systems. An operational task roots achieved through localized chemical sensory loops. This subterranean surveillance relies entirely on exudate fingerprinting. Roots continuously shed a complex cocktail of chemicals into the local soil matrix, including proteins, organic acids, and secondary metabolites. The cells of the root cap or calyptra continuously sample these adjacent chemical gradients. By decoding the specific ratios and molecular patterns of these compounds, a plant determines whether an encroaching root belongs to its own structure, a genetic twin, or a competing non-kin species. When a plant detects a sibling or its own cloned roots, it initiates a kinship shunt, downregulating its lateral root elongation. This cooperative response prevents self-strangling, optimizes mutual root spacing, and conserves precious metabolic energy. However, when a root apex identifies a non-kin competitor, its internal signaling shifts immediately. The plant ramps up the production of growth hormones such as auxins and cytokinins, launching a rapid lateral expansion designed to intercept the intruder and block it from accessing local resource patches. Tactical maneuvers, interception, proliferation, and mechanical strangling. Once a competitor is detected, root systems execute physical maneuvers designed to cut off the enemy's resource lines and claim the local soil volume. This strategy operates primarily through precision nutrient foraging, turning resource acquisition into a high-speed race. Highly aggressive plants do not expand their root architectures randomly. Instead, they deploy dense, highly branched networks of fine feeder root hairs directly into the growth paths of adjacent plants. By completely surrounding a patch of nitrogen, phosphorus, or water before a competitor's root can reach it, the aggressive plant forms a dense biological barrier. This overgrowth sucks up all available resources, leaving the rival root stranded in an exhausted nutrient desert and causing it to wither from localized starvation. This competitive exclusion scales up to the mechanical encirclement and constriction, where certain invasive or dominant species use physical force to suppress rivals. Their root wrap around competing root structures in tight, spiraling loops. As these encircling roots absorb energy, they undergo secondary thickening through lignification, hardening into a rigid wooden frame. This structural growth exerts immense physical pressure on the rival root, crushing its delicate vascular tissues, specifically the xylem and phloem. This mechanical constriction chokes off the host's ability to transport water upward and translocate sugars downward, systematically killing the rival root systems from the tip up. Empirical case studies, root battles in action. The real-world execution of these subterranean combat strategies is vividly captured in the long-term ecological field studies. The spotted knapweed, Centaurea stowweed, serves as a premier example of how invasive plants deploy targeted root tactics to displace native vegetation. The roots of the spotted knapweed actively pump out catechin, a powerful phytotoxic chemical, into the rhizosphere. As the knapweed roots grow rapidly into native root territory, the accumulating catechin triggers a wave of oxidative stress within the cells of native plant roots, forcing them to self-destruct. This biochemical attack melts away the native spot system, allowing the knapweed to quickly take over the empty soil volume and secure absolute access to groundwater. This intense competition can pull plants into an evolutionary trap, demonstrating by the common bean, Fasciolosis vulgaris, under conditions of high-density-driven overinvestment. When individual bean plants are grown in close proximity with non-kin competitors, they enter a destructive evolutionary feedback loop. Even if a nutrient patch is already completely full, the plants will divert up to 30% more of their fixed sugars away from leaf and seed production just to grow overdense, combative mass of roots. This root explosion is designed purely to crowd out the neighbor, demonstrating that plants will willingly sacrifice their own reproductive output to win a territorial subterranean war. This unyielding, energy-draining feedback loop provides a powerful botanical parallel to the behavior of the resource-heavy Homo colossus.
We negotiate with bombs. You have a choice as we loiter over the top of Tehran, as the president talked about your future. The president has made it clear that you will not have a nuclear weapon. The War Department agrees. Our job is to ensure that. And so we're keeping our hand on that throttle as long as hard as is necessary.